Hello friends, this is Dr. Preeti Srivastav and I welcome you all to my YouTube channel Life Science by Dr. Preeti Srivastav. Today is a great day. The Royal Academy of Sciences has announced the Nobel Prize in Chemistry for 2020, which is backed jointly by two women scientists, Emmanuelle and Jennifer, for the development of a method for genome editing. As this subject is very close to my heart and I am doing great efforts for spreading the awareness and spreading this particular subject to the people. So I am very excited and happy to share this particular finding, this particular discovery with all of you. It may be called as using the genetic scissors. Yes, now we can cut, edit, rearrange and modify our genomic structure. Genomic structure or DNA. DNA which is called the code of life. As all of you know that our body is comprised of trillion of microscopic cells and each and every cell is having this genetic material called DNA. This DNA is composed of thousands and thousands of nucleotides and this long string now can be cut at specific locations with the help of this particular discovery. So, the CRISPR-Cas9 technology is a major breakthrough in gene technology. However, the gene therapy, recombinant DNA technology, and different genetic engineering technologies are there since 1970. Then what's new with this technology? As it is much cheaper, it is very safe, it's less time consuming, and it's very quick, very fast, and can be easily maintained in most of the biochemistry and genetic labs. So we'll know something about what it is actually CRISPR. What does it mean? The full form of CRISPR is clustered regularly interspaced short palindromic repeats. And Cas9 is a protein which is associated with this particular DNA fragment. So in simple terms, it is a specific kind of DNA present along with this particular protein Cas9. Both of these and they are associated with some kind of RNA. They make a kind of a biological or you can say molecular or genetic scissor kind of thing which can cut the DNA at particular or very specific sites. Now how it can be useful? If it can cut the DNA, it can be used to modify our crops in plant breeding, in so many genetic diseases. We can cut those mutated or defective genes. It is very useful for the mankind. And this is very interesting thing that this particular mechanism, that is CRISPR-Cas9, this genetic scissor mechanism is a natural mechanism found inside the bacteria. It is the part of their adaptive immune system which they use against the viral infection and to memorize it that whenever in future if the same virus attacks, they can defend themselves. So this particular mechanism was discovered by one of these scientists and then they worked on it and they planned to reprogram it in the test tube. Now coming to the application part of this particular technology, it has wide application. Wherever there is any living organism, plant, animal, microbe, wherever there is the presence of DNA, this is very useful and helpful technology. So in terms of food and livestock modification, it can be very useful. It is very useful in plant breeding and animal husbandry. Apart from this, the major breakthrough can be in the gene therapy. It can be very useful for the cure of point mutation cause genetic diseases like sickle cell anemia or other genetic diseases which otherwise have no clue, no cure, no permanent cure is there till date. But with the help of this technology, we are very hopeful that there would be a permanent cure for the genetic diseases. There are some other areas like human germline and designer organisms where it can be very helpful for the production of designer babies or superhuman. But at the same time, we have to consider many societal and ethical issues associated with all these parameters. The whole scientific community is looking forward as this particular CRISPR-Cas9 technology would prove very helpful for the cure for genetic diseases. On my end, I am also creating awareness about these genetic diseases and how we can prevent them. Till date, all of us know that there is no permanent cure, but we are very hopeful that this technology may prove to be very successful to cure the genetic diseases. This is my YouTube channel and this is the link for the genetic diseases related videos. Kindly go through. Stay tuned for detailed version of CRISPR. Next videos will comprise of the molecular level technology of this particular CRISPR-Cas9 system. So stay tuned and thank you.